Malaria is caused by a pathogenic organism called Plasmodium, and 1.2 billion people live daily with a high risk of contracting malaria. A malaria infection begins when a mosquito carrying Plasmodium parasites probes the skin of a human for a blood meal and injects tens to hundreds of Plasmodium forms known as the sporozoite. The sporozoites leave the bite site, enter the bloodstream, and are whisked away to the liver, where each sporozoite will find a single liver cell to infect. Over the next 7 to 10 days, each parasite will transform, grow, and divide into tens of thousands of new parasites called merozoites. Meanwhile, the infected person remains without symptoms. When the merozoites are mature, the infected liver cells burst and release them into the blood. Each merozoite will then infect and multiply into dozens of new merozoites within red blood cells and burst out to infect more red blood cells. The infection cycle rapidly expands the parasite number into the billions, which causes crippling fevers, body pain, and symptoms in the 200 to 300 million people infected with malaria each year. Sadly, 600,000 of those infected, mostly pregnant women and children, will die. So how do we stop this complex cycle of transmission, disease, and death? By turning the parasite's strengths into its greatest weakness. Scientists at the Center for Infectious Disease Research analyzed all the genes in the malaria parasite using systems biology techniques and identified genes that are essential for the parasite to grow and multiply in liver cells. They then removed these genes using genetic engineering, creating a weakened parasite called a genetically attenuated parasite. This weakened parasite can then be purified and bottled as a vaccine, in the same way that weakened versions of the polio virus have been used to nearly rid the world of polio. When injected, the genetically attenuated parasites are able to travel to the liver and infect liver cells just like a normal parasite. But instead of dividing and progressing to the blood, they get stuck and die in the liver, alerting the immune system to their presence. This generates antibodies and T cells trained and stationed in the liver to recognize and kill any invading malaria parasite in the future. Now, in a vaccinated person, when an infectious mosquito bites and injects sporozoites, they can be recognized by awaiting antibodies and blocked from invading a liver cell. And those that do manage to infect a liver cell are rapidly destroyed by patrolling T cells. This immune response stops the parasite from ever making it to the blood, preventing disease, death, and further spread of the parasite by mosquitoes. This kind of highly protective vaccine provides our best hope for a world that is free of malaria. Center for Infectious Disease Research. People, science, hope.